Welcome back, Year 12s. We're looking at adaptive immunity. In this case, we're looking at antibodies specifically, also known as immunoglobulins. So antibodies are released by the plasma cells, and these, of course, are derived from B lymphocytes that are stimulated. And of course, antibodies are proteins, just like any other protein. So they have certain segments to them. They have different bonds. All right, and in this case, they have a light chain, a heavy chain, okay, that we need to be aware of, and the two active sites. Okay, so they're composed of 4 polypeptides, so 2 proteins, and on the outside, which are the light chain, and then 2 proteins on the inside, which is the heavy chain there. Okay, and they're connected by disulfide bonds, and each antibody has a antigen binding site, which is a variable region. So that changes and is specific to a different pathogen's antigens. So these regions come together to form 2 identical antigen binding sites, okay, unless of course there's a specific antibody that we're looking at. And as there's 2 antigen binding sites, an antibody can bind to 2 different pathogens at once, effectively sticking them together, all right, which is then used by many antibodies together to create a certain mechanism of destruction for those pathogens. So they're also known as immunoglobulins antibodies, okay, and there's 5 different types that we need to be aware of. There's IgG, there's IgE, there's IgD, there's IgA, and IgM as well. All right, VCAR probably won't expect you to know in detail what each type of antibody does. You are, however, expected to know the general structure of the antibody. Okay, so that previous slide we looked at. So how do antibodies actually destroy the pathogens? So there are multiple ways antibodies destroy and inhibit pathogens, the most notable being what we call agglutination. So that's that sticking together process. All right, antibodies bind together with antigens, and they also bind with pathogens, and they create like a giant web, essentially. Okay, so that's this number 4 here. When that giant web is formed of the pathogens' antibodies, okay, stuff like natural killer cells will come along, all right, and stuff like phagocytes will come along and engulf them because they will signal them with cytokines and other molecules. Okay, there is a few other ones we need to be aware of. Okay, so like bacteriolysis, breaking up the bacterial cell wall, which destroys the cell. Opsonization, which essentially just sticks to it to flag it for a phagocyte. Alrighty, neutralization as well. And yeah, that's what we really need to be aware of. Okay, but the main one here is agglutination. Thanks for listening to our quick video today. Have a nice day.